Well, welcome everyone. I'm Joanne Geer. I'm the Executive Director for the Westchester Biotech Project. And today is our December meeting on advancing microbiome science to clinical practice. This is a program of the Consortium on Translational Research in the Microbiome, which is a collaboration between Westchester Biotech Project and the Institute for Life Science Entrepreneurship, which is at Kean University in New Jersey. We have a couple of speakers today, and after they do their short presentations, we'll have a round table. So I'll be inviting each of you to introduce yourself, ask any questions, make any comments. And what's nice about these sessions is that we very often end up with a theme by the end of some idea that people think is worth exploring further, and that gives us ideas for the next sessions. We have, if you registered recently, you will see that we have booked our dates all the way through August. We were hoping last spring in April to have an in-person conference, and obviously that couldn't happen. So we are hoping, there's big fingers crossed, we realize it may not be able to happen, but we're thinking of trying to do an in-person session in the summer. Just to have the fun of looking forward to it. But these monthly sessions will help build up to that. And anyone who is interested in speaking, being part of planning, being part of steering where this goes, we certainly invite you to speak up. I'm first going to I'm just going to tell you a little bit about our organization. So we're going to be hearing from Nini Fan, Karen Woodward in a moment. So the Westchester Biotech Project is really all about bringing together researchers, data scientists, and engineers, and those who love them. So it's all about cross-fertilization across all the different silos of biomedical research. We have two major pillars. One is our career consortium, which includes pedagogy programs, includes building your first laboratory, includes things about grant writing and grant seeking, all different aspects of being a scientist and building a scientific career, a science career. And then the other pillar, which is launching as we speak, is the Westchester Data Analytics Hub, which will provide an infrastructure for collaborating across the different data silos, from discovery to preclinical to clinical to patient care to population health. So our first element will be around population health, and that will focus on both training data scientists with classwork and internships, and training managers to work with data scientists all around biomedical data. So we'll be saying more in the future about our plans for 2021, which we're basically putting together right now, but got some wonderful things in the works. We are grateful to have many different entities where we have been able to collaborate. Some of them have given us funding, some of them have hosted us for conferences or been part of steering committees and so on. We're very grateful. We started about 3 years ago, so we're very happy about the relationships we've been able to build. And so first I want to actually introduce Jessica Dunn, who will tell you a little bit about our consortium and about herself. Go ahead, Jessica.
Thanks, Joanne, for the introduction. So I've been working with Joanne and Keith Bostian, who's unable to join us today, and Raj Shetty, who is a little under the weather today, so he's here and not speaking, for the past couple of years on developing this Center for Translational Microbiome Research. And the real goal of this was to really bring together people from different parts of the microbiome research to figure out how we could really advance microbiome science. As Joanne talked about, we really had this wonderful in-person meeting planned for April, where it was really about how do we drive these early research studies and how do we get them to inflection points, and what are the barriers to really delivering therapies to people in the microbiome space. And so, like I said, we've been working together for the past couple of years on this. And I don't want to spend too much time, but really the focus here, and as part of what we should be thinking about doing today during the discussion period, and you'll hear from our wonderful two speakers shortly, is really about their work and what they're doing. But hopefully you'll hear about some of the challenges and opportunities in microbiome research. I spent 12 years at JDRF before moving in June to Janssen. And with a focus on type 1 diabetes research. And, we really think about how the microbiome is broadly applicable, and therapeutics based on the microbiome are probably broadly applicable across multiple disease states. And how do we leverage what we're learning in one model system to really think and talk about other diseases and other model systems, and what are the gaps both in terms of commercialization and marketing and manufacturing, all of those things. And so hopefully we'll be able to sometime maybe in late 2022 or 2021 to bring that wonderful meeting back to life. So I hope you'll enjoy our speakers. I'm going to punt it back to Joanne for the introduction, but I really encourage everybody to actively participate. Think about how the two speakers that we have today and how their work, and maybe it's a different, again, disease or model system than you're working in, but how can you leverage what they're working on for your own research, and where can we also think about building collaborations across multiple disciplines to really advance the science. I said a couple of years ago, and I'll stop talking after this, but I think the microbiome hit really maybe about 10 years ago at this point in such a splash. It was almost like what happened with genetics a decade before that there was such a splash in how genetics and how genetic research was going to change the world, and then there was a lull, and now we're seeing this renewed interest in genetics and how therapeutics, specifically targeting genetics, may be useful. So I feel like we're in a little bit of that lull period with the microbiome, but hopefully you'll be convinced otherwise by our two wonderful speakers here today. And I guess I would just implore you guys all on the phone to make sure that we are really driving forward, so that there's not a loss of interest in this field. I think there's a lot of power in our microbiome, and how we can leverage that. And again, I think our two speakers will really talk about some of the wonderful and foundational and really outstanding work that they are doing within the context of their organizations. So please participate. It's what makes these more enjoyable for all of us. And so with that, I will pass it back to Joanne to speak about our wonderful panel here today.
Well, thank you, Jessica. And so I'm going to stop sharing so we can have bigger pictures of everybody. And so we're first going to hear from Nini Fan. And let's see, we need to, Matt, if you can hear me, would you make Nini a co-host? And Nini, you need to unmute yourself. And
I think I'm good to share right now, so I'm going to share the screen.
Great.
And then, can you guys see it?
There we go. Off you go. Thank you.
The presenter view. Okay. Awesome. Okay. Hi, everybody. Thanks for being here today. I am Nini Fan, a microbiome researcher, and I studied and specialized in the microbiome research, and particularly in the genomic sequencing area and bioinformatics. And I'm also currently the founder and CEO of MaMome. Today, I would like to talk about our research and science, and where are we in the commercialization path, and some of the challenges we have reached. Yeah. So
I have someone.
So the scientific community has been found that the symbiotic relationship between human body and then its diverse microbiome is the key to understanding and improve nutrition, diet, and health. So data increasingly show that microbiome could be altered and influence the health in human more ways than previously than we know at this era. And diet is one of the most important factors that can be used and regulate a maternal microbiome. And it also has directly a lot of link to a serious medical conditions such as gestational diabetes and hypertension.
Someone in the Asia Pacific region to help manage my Asia Pacific distributor. So just document what it.
Yeah. So I started, I don't know, there's a back of the voice.
And you couldn't live without, what'd you find out you could live?
Let's see, can we, that wasn't you, Nini, right? That's someone else?
It was, yeah, I'm just kind of, it's kind of like weird, the voice coming back.
Yeah, it sounds almost like it was our sound check recording coming back to us.
Yeah, so I wasn't sure. Yeah, I'll keep going.
I'll just say that if you're not speaking, please mute yourself and hopefully we're going to be okay.
Yeah,
thank you.
I'll keep going.
Yes,
so a little bit background of myself and then how I started all of this. So I studied in Dr. Jimir Molina's lab, which is from Long Island University, Brooklyn campus. In my graduate school years since 2017, we were focusing on the study on the analytic and the effect of natural products such as grape-induced colitis to the gut microbiome by using the 16S ribosomal gene for next generation sequencing.
You couldn't live without, what you find out you could live
in the
without.
lab that we picked 31 mouse out of 4 groups, which is out of 128 mouse in 16 groups. And we were mostly focusing on the sequencing techniques aspect, which is the 16S ribosomal gene. And approximately there's about 50 base pair long and consists about 9 variable regions, which is V1 to V9 that include conservative regions that crucially represent the ribosomal functions. So in the structure setting that we utilize the variable regions, which is suitable as the phylogenetic markers to differentiate between the species and strain, while we use the conservative region to serve as a binding site for the primers to design this amplicon sequencing variable regions for PCR application.
Yeah, and just
so in this
put a
research,
pin in the air because holy smoke.
we can across that realize the broader impact of microbiomes and their host.
A college age daughter finishing up her finals at home, and I have a high school son at home, and my husband's also been working from home since March. So
recording.
Oh, I hear what that is. Matt, we really need that to
well, if anybody needs some comic relief.
This is coming from Nini's microphone at the moment. Something on that end is.
Well, what we're hearing is what we thought.
That was very funny.
If Nini were to mute the mic, that weird voice would disappear, according to Zoom.
Yeah, and there's.
I don't know, understand it, Nini. I can see everybody.
Group, but I'm not a big.
Huh.
Yeah. If I mute myself, would that be better?
Well, but then we wouldn't be able to hear you, would we?
Yeah.
Unless you want
to. Okay. I think now it's good, right?
It seems to be okay. So I'm going to suggest if you hear it again, just pause so we can actually hear you because it seems to be intermittent. I'm sorry about that.
Yeah, I'm sorry about it too. It seems like it's the recording from the beginning. Yeah.
Okay.
So I'll continue. So from the research that we realized that the broader impact of the microbiome, the microorganisms with their host. So fundamentally, this symbiotic relationship between the host and microbiome can be really influenced in many ways, including the disease and especially in the earlier colonized gut microbes, which is very interesting. And then it affects the whole lifespan. So from there,
the musical. That was very funny. You saw it was
yeah. I guess once I switched,
it was just what I needed.
Sorry, I muted you by accident, Nini. Matt, can you put in the call-in phone number?
For musical.
into
2020, the Hallmark movie, and
Colin
so watched the Hallmark movie one the other day.
We should be okay.
Okay. I apologize for this. Yeah. No idea what happened.
Yeah, I'm hoping, but
Yeah,
right.
our earlier conversation, but we weren't recording then.
I'm trying to change the format of the PowerPoint so the sound doesn't come back, so I can just just read from the PowerPoint. My apologies. So should I continue?
Yeah, let's continue while it's working. Yeah.
Okay. I guess I'll use this format because I feel like this doesn't have the sound. It could be that recorded.
Oh.
So from here, then from the previous research that we discovered and realized the importance, then we realized that starting with a pregnancy, which is all the way in the beginning of lifespan, is so much important to understand how earlier a gut microbiome was formed and transformed it from the mothers to the infant. So in that case, we decided to build a data-driven analytic database for pregnancy to track and identify the microbiota in healthy and unhealthy pregnancies. So because of that, we started our proof of concept technology platform by getting IRB university IRB approval, which is required and to study hundreds to thousands of stool samples from the expecting mothers throughout each single trimesters. In our study, we also compare the pregnant healthy mother, which is the host, with the unhealthy pregnant mothers group. Different groups would follow by different types of a diet by recommending it. And there's also the random group of a diet that's also provided to the healthy and unhealthy mothers for the future comparison. How we demonstrated our technology is to collect the sample from the OBGYN's office, which is once pregnant women realize they getting pregnant the first trimester, when they go to the doctors in 2 weeks, they will start getting the sample. So here we have, once we collect the sample, we do a series of next generation sequencing by using actually Shoreline Biome's next generation sequencing kit to run our data analytic, which is our unique method and algorithms that we patented for MaMome. And essentially, this could come out as a clinical report to show the clinicians and end users, which is the mothers, in each single trimester to make them understand how their diet and the microbiome will work. In the technology, we really want to emphasize and develop this framework of what is the balanced microbiome. So we want to establish the healthy pregnant mother's profile first, and from there that we derive and mark what is the standard or balanced microbiome to understand the unhealthy pregnant mother's outcome from the gut microbiome. So our work in here, you can see a little bit the RAS sequences, which is the microbiome sequences, can come in, and we do a series of bioinformatic analytics and to essentially fit into our proprietary AI system with awful, and essentially there with enough of the good data, we should be able to see what is the high risk microbiome profile from the enough of the data. Then this is part of the earliest batch results from our clinical trial that started this year. They are particularly focusing on the healthy trimester 2 patients and unhealthy trimester 2 and healthy trimester 3 and healthy trimester 3. We started that because these high-risk complications such as gestational diabetes and preeclampsia, women don't really get diagnosis of them until trimester 2 of 20 weeks to 27 weeks. So essentially that will be a good, so that is one of the approach that we want to see in trimester 2 and trimester 3, if there's any link in between these high-risk pregnancies. And from our the data, we identify at least 680 unique identified strain from just this very little cohort. And also 48% of the strain are not identified from the microbial database, which is very, very mind-blowing to me. And here is a brief pie chart that prove that if we can get an earlier detection, we can actually lower the preterm birth, which is preeclampsia and gestational diabetes directly affect for. So we want to take the approach that if we can start sequencing microbial for the pregnant mother in trimester 1 in between 0 to 12 weeks, we can have a better sense and understanding what kind of bacterial indicator that will be presented in there. And here is more visualization pie chart. So in here, we have the comparisons of a healthy pregnancy profile and unhealthy pregnancy profile. And this profile is, this patient is picked, especially have a diagnosis for preeclampsia already. And then you can see the rest of the abundance representative is mostly the same, but with this particular strand, there's different percentage, which is very different, large right here. And then we also compare them with the healthy pregnancy trimester 1 biota. And then also we have a control group as a healthy non-pregnant woman to see the differences of a pregnant woman and non-pregnant woman's differences, and not just from trimester 1 to 3 and advancing. So pretty much that's how far that we gone through, but our clinical trial is still going. Due to the pandemic, it is seriously slowed down due to, the doctor's office is closing down for, half of the time. And particularly for pregnant women, they're super careful and they don't want to leave the house. And with this typical type of the nutrition clinical trial and intervention is very, very hard and complicated for a woman who's currently pregnant that go out to grab food and following the restriction diet. So these are kind of like the main challenges that we're reaching while we are conducting this clinical trial. We're not sure at this point, like how long we should keep really the enrollment windows gonna be last. And realistically, once we start running it, we realize that the drop-off rate is actually really high. So there should be other things to be implemented, and also to really see what is the effect of diet. We need to understand how we exactly collect the data, if the data is actually monetized in the regular way. So that is the majority of the challenges that we're reaching, and hopefully the end of it, we will have all the patients recruited and then do a full genome sequencing run to do the comparisons. Yeah, and here's my team. Right now I have me, and then here's my PI. We have marketing and nutritionist, and also most importantly, we have artificial intelligence and data scientists on board to really help out to understand how we can do supervised and unsupervised learning in this large and complex data. Here is, this is just a last graph, have a map of how a pregnancy journey is and how standard of care is currently is. In trimester 0, trimester 1, there's no certain detections. And then often with this journey end up people with the high risk pregnancy will end up preterm and in the hospital with a lot of hospital bills. So we wanted to create a maternal microbiome profile timeline to assess and help with the overall standard of care, so we can decrease the maternal health impact overall. That's pretty much it. Thank you. Sorry about the sound system today. It's very odd.
We learn something new about Zoom every day, don't we? Well, thank you very much, Nini. And that was terrific. We're going to go right ahead to Karen Woodward and let you share your story. You can unmute. And let's see. Matt, have we made Karen a co-host so she can show her screen?
I think yeah, I need to be a co-host, I guess.
All right.
You should be good to go.
Well, there you go. Thank you.
Okay, can everyone see my screen?
Yes, we can.
All right, let me put this on slideshow in a minute.
On the bottom, there's a thingy.
Yeah, I can't.
Over toward the right, over toward the right, way on the bottom on the right. That's it. Oop, not that.
Yeah,
that.
there we go.
Yeah.
There we go. Well, thank you, everyone. I'm going to talk a little bit about Shoreline Biome and my role, as well as sort of how we started, what we're focusing on, and some of the things we've learned since we were founded. So I'm Karen Woodward. I'm the Vice President of Business Development of Shoreline Biome. I've been with them about 2 years. That's about the time that they decided they're ready to launch product, which I joined with our new CEO. But I've actually known them since their inception. They are located in the Yukon Incubator in Farmington, Connecticut, which I was formerly a part of the University Technology Transfer Team. So I got to know them very well. And then they convinced me that they're ready to launch product, that I should jump back out of academics since the startup. And so I haven't looked back since. So they were founded in about 2014 in Farmington, Connecticut, with 2 former Roche 454 business folks who were very instrumental in the launch of the first long-read sequencing platform. And so at the time when Roche decided to move everything to California and not really play in that space anymore, they had just started working with leveraging long-read sequencing in the microbiome space, and decided to take that upon themselves to really drive that forward. So we've had a Series A fund. We're in the process of raising Series B, launch product about 2 years ago, which as you can see, about a year of that has been within COVID. So it's been an interesting challenge. I'm glad we got a head start, but now we have a lot of catching up to do in 2021. We have US direct sales. That's myself and 1 other person. So we're lean and mean. And we have 8 non-US distributors around the globe, and hope to expand that next year. So the premise was really to, again, push next generation sequencing into the microbiome, leveraging more of the long read sequencing, but also try to solve some of the other problems that the team was starting to see and folks that wanted to really maximize, be able to use long read sequencing. So we now have a team with experience from a variety of different areas that have all come together to kind of address this from the perspective of our customer base, which is really a mixture of translational scientists and academics, where majority of the microbiome research still sits, but also some of our customers are microbiome therapeutics companies, pharmaceutical companies, ag companies, as well as service labs. So that's one of the challenges, is it's quite disparate. There's a lot of different customers in the microbiome space, but that actually is quite a benefit, because each one of them is kind of poised, ready to grow at its own pace. So we're focused on really getting them an end-to-end solution, and really focus on the ability to really look down to the strain level of the bacteria, which has really been a challenge to be cost effective and democratized within the space. So I won't go too much into the market opportunity, other than that, we firmly believe that the microbiome world is growing at a rate at which people, I don't think we're expecting. And as I would agree that one of the things that I've learned is this has sort of been a rebirth of what I would say a little bit of stall in the genomic space. I come from the genomic space, so it was kind of nice to see this sort of growing again. And it's really, despite COVID, been a landmark time for microbiome, with investors betting big on some big companies, as well as grant funding going into some pretty significant areas, including but not limiting to infectious disease, oncology, as well as the ag space. This sort of reiterates a bit what I have said, but I don't think anyone can argue that the the amount of grant dollars that are going into microbiome and the amount of publications that are coming out are growing at a rate that I cannot keep up with them. That's one of my challenges. I try to keep up with what's going on, and it's really difficult. And then it's really hard to keep up with who's doing what and focusing. I had someone ask me recently, so which diseases do you think the microbiome is going to play a key role in? I think the answer is that nobody really knows, but pretty much every disease out there, disease area is being looked at from the microbiome perspective. How that'll all land, I don't think anyone knows, but I don't think any disease area in human health is sort of untouched by the microbiome research right now, which I think is pretty unprecedented. And so it's really fueling a lot of research and a lot of answers that are coming out. And there's a lot, I didn't have enough room to put all the logos for all the companies that in some way are working on microbiome, either directly through a therapeutic intervention development, or just looking through their current clinical studies that aren't really microbiome focused to understand how the microbiome of the patient or the clinical study participant, how their microbiome is either being affected by the intervention, or if they can predict either the safety of their intervention or the efficacy by the microbiome at some level. And again, keeping up with the clinical studies is really hard as well. So it's a great time to be a scientist. It is a challenging time to be a startup and figure out how you play in that space. So our areas, and again, this is a problem that we're trying to work with our customers to solve, that the methods out there for microbiome research, some fall short of meeting the needs for both being able to discover novel microbes, but also identify things and distinguish them on a strain level of what is known out there. And we absolutely know this is going to be essential for clinical and translational research. So there's a lot of things out there that have sort of brought up a lot of questions, but we're still kind of limited in answers and being able to look at things down to the strain level and also not miss the things that are novel. What we have found in our customers report, most of the microbiome is still unknown and uncharacterized. And so, I was looking at the slide this morning actually again, and I think this is probably not as up to date, but I think the story is really that, if you focus too much on the knowns, you're going to miss stuff. But trying to find the unknowns and figure out what they're doing, what they are, how to classify them, and then especially what their function is not easy. But what we're finding is a lot of the research, when they identify an unknown, it is the unknown that seems to play a key role. And so the question is, do they focus on sort of what's known, or do they sort of divert and spend a little bit more time reaching on things that may not be well known? And this is definitely true in areas not only in the clinical space for the gut microbiome, but expands even past that into things like wastewater. So not to divert a bit, but everyone I think is aware that the COVID pandemic has really opened up another question about wastewater. People have been looking at the microbiome and wastewater for quite some time now. What we find is most of the things in wastewater are not known, which is very interesting. But people are actually now trying to track COVID in wastewater. So, and that is going to be interesting and actually will open up some new areas of even the microbiome to figure out, can we track things and detect them early in wastewater, as well as looking at things in the stool samples, the skin. We've seen every sample under the sun come through. So there's not even a sample untouched where people are not trying to look at the microbiome. So there's a couple of key questions here that have arisen with this microbiome revolution, that if we focus on things that require culture, we're missing a lot. So culture independent methods have become extremely important in the microbiome. Traditional methods, I can't see my slide here, of lysis can be very biased, and we've done a lot of work in this area. And that's still, I think there's no standard methods to make sure if you can't break open the cell and get the stuff out, you're never going to detect it. Full-length 16S still can't distinguish most strains. When you're trying to do shotgun methods, host DNA confounds the data. So depending on which sample you're taking, if you're trying to look at the bacteria and you're taking off skin, the majority of what you're looking at is human DNA, and it's hard to pick apart the two. These are things most microbiome researchers deal with on a daily basis. If you have to assemble a strain, you can miss things that are novel. And long-read NGS really does offer high-resolution data, but still needs democratization, low-cost methods, and standardization to make it to the clinic. And it's going to be interesting to see how that evolves. So I wasn't going to spend too much time on our data, but I just wanted to pick one example at which why we are focusing on trying to distinguish things at the strain level using an amplicon-based method, and that's what we call our strain ID. And when I say strain ID, I mean everything from lysing the sample to an interesting long amplicon that expands past the usual 16S, as well as a novel database, because all of those pieces have to come together. And one example is looking at something as simple but yet important as C. diff. If you even attempt to try to map something like C. diff in a standard 16S database, you just can't pick the strains apart. And here, knowing the strain of C. diff is going to really impact any decision you make, because people think bad things about C. diff all the time, but really depends on the strain. Some things can be resistant. Some strains you can be infecting and shedding spores to other people before you even know it, and other strains have shown to be protective. So really kind of distilling down to specific strains of microbes is quite the challenge and something that we're focused on. So we've developed this novel assay, which is an extra long amplicon, which allows our customers and the researchers to leverage the ease of use of next generation sequencing on the long read platforms, whether it be PacBio or the Oxford Nanopore, and be able to look at things and not have to have a preconceived notion of what's in there, because you'll pick up a known and an unknown and not have to assemble it before you go to something like shotgun sequencing. And the clinical translational medicine folks that we're working with really find this important, because if they can't understand the difference of the strains, they can pretty much be chasing the wrong things for too long. And for things, if you're going to get to pathogen detection, everyone knows that you kind of have to understand the strains. And 16S traditionally has not been able to do that. So we're hoping to kind of transform that and work with some of our early access customers, either going to more focused assays or even eventually to the clinic, leveraging some of the new next-gen technologies and assay development platforms, such as Nini and her team, to make sure that they're getting the best strain level resolution out of their data. E. coli is a very common case study, which we use all the time, but it's not the only pathogen out there to look at. So I just wanted to kind of put this out there. One of our challenges, which I'll talk about on the next slide, is that there is a lot of methods out there for doing microbiome work. And on a daily basis, I talk to each folk that has their favorite method, and they all have their pros and cons. So kind of understanding from a customer perspective what their question is and what things they want to determine can be very important, and kind of figuring out which solution is best for them, because they each have their pros and cons. So whether or not you need very high bacterial coverage, whether or not you have a sample that has a high amount of host DNA, and whether or not you need species resolution or a genus answer is okay, will kind of depend on whether you can go to traditional 16S sequencing. We have kits for those as well. Whether shotgun sequencing is really the most important thing for you to do, or you can work with us on our what we call our 16S ITS 23S sequencing, which is strain level but at an amplicon. Detection of unknowns is one thing that we find is quite valuable, that's quite difficult with the other methods. And also informatics is key, because there is a lot of informatics that may be needed for the different methods. And if a user wants to do that themselves, they either have to figure out if they're going to use sort of a free version of software, do they need to program, do they need to assemble data? And that's something that bringing those types of solutions to a clinic kind of have to be resolved and made easy for a clinician to do before this can really move forward. Sample throughput and costs do vary. So depending on how many samples you need to do and what your sensitivity for cost per sample will dictate sort of which side of this, this table you lie on, and sort of working through all of this. And then in some customers going straight to a PCR assay, if they have a couple of key microbes, Next generation sequencing isn't going to be the right solution, but making sure they understand how to get there and the research that needs to get there is extremely important. And those are the things we talk to our customers about daily. So my last slide is really, there's a lot of challenges in this market and a lot of things that we've learned in the last 2 years. You know, and these are just there's challenges, and I list these as opportunities because these are things that people are working on today, and it's not an extensive list. But things like biobanks are becoming quite necessary for some of the studies, and there isn't a lot out there, but the pharma folks I know have told me, especially in times like now where you can't collect clinical samples, they've kind of tried to lean on biobanks to continue their studies. And they're not quite there yet. And I don't think anyone's really figured out the best way to standardize collection and storage, how the sample collection impacts data, and they're not as easily accessible. I mentioned the computational challenges. These data sets are now becoming extremely large. The methods are varied, and it's really hard to compare across studies. Standards are definitely needed, and there are groups that are working on that. The question of whether this is a cause and effect, can it be an intervention? The impact of the microbiome, as we know, is multifactorial. Even what you eat on a daily basis is going to affect the drug that you take. So how do you determine all those multifactorial approaches on the data? So there's really going to be a lot of computational power put behind this. And I think it's going to result in some really key inventions. There are translational gaps. There's not a lot of good preclinical models. No one knows what the normal microbiome is. I talk to the regulatory folks and the pharma all the time. You know, these are going to, if you're going to develop a microbiome therapeutic, these are live biotherapeutics in some cases, and they really have to drive some process for the GMP productions as well as the approval. And formulations for these is not necessarily going to be cheap, and you have to know which strain is in there and be guaranteed that there's not some weird strain creeping into your biotherapeutic. We've learned that, you know, samples are highly variable and unique, and every customer has a very unique sample. Even skin from one lab and skin from another don't behave the same way. And so those are the things you have to understand when you're designing a project. Collection is also not standard. There's a lot of kits out there, but there's no standard way, and how you collect your sample is definitely going to impact your data. More mock standards are needed. The ones that are out there work, but they're really fickle. And if you even handle them inappropriately, they tend to change because these bacteria tend to lyse sometimes very easily. And so we found even the centrifugation of a mock microbiome sample will affect its outcome. Analysis, everybody's doing different kinds of analysis. A lot of it's homebrew. And comparing, it's difficult to compare data sets. This is a fast technology area. Keeping up is a challenge, but it will solve a lot of problems. We work a lot with our customers on experimental design now, more than I think the founders ever thought we would, because experimental design is key. And the hardest thing is, you know, this is a risky change for our customers, and crossover studies take time. And so to convince a customer to change how they do what they do when they've been doing it in the Human Microbiome Project for a couple of years now is really hard. So data, and more data and publications will help to drive the customers. I think ability to risk change. I just put a quote here on the bottom, which I read the other day, which I think kind of sums it up. And it's really from Harvard, where it says, the microbiome research is really entering into an exciting new phase where we can start tying things together to a firmer grasp on the mechanisms of what's happening to microbes in humans over time and how it relates to disease. And I think that it kind of says although a lot is happening, we're probably still at the infancy of this growth curve. And so I think there's a lot of interesting things to come. And that's it.
Well, thank you, Karen. Thank you very much. I'm going to ask the first couple of questions, and I just want to let folks know that when we get into the roundtable section here, that we will post this first section and the part where I'm speaking to the speakers on our YouTube channel. The rest of the conversation we will not include unless you say something so wonderful we'd like to include it, and then we'll ask you for your permission to do that. Karen, I'm going to start with you, and then Nini. Karen, because you mentioned it, excuse me, I'd like to hear about your experiences with the FDA and what kind of challenges or openness you're seeing for new types of microbiome products. Karen first.
Yeah, there have been, I would say, pretty high level and mostly through our collaborators and customers. And I think what they're saying is they're kind of looking for some folks to take the lead. And there are some standards that they're asking for. And one of the challenges that I think they're pushing on the microbiome therapeutics companies that we've heard is the ability to say, if you're for a consortium drug, for example, what data are you going to provide me to convince me only those 3 microbes are there? And then the drug developer will say, well, what level of sensitivity do you know I need to show that? And then they will push back and say, well, you need to sort of define that. The standard for that is qPCR for the most part. So I think that's an area where they're waiting for someone to kind of show the data, not being clear what data they want. And so I think, you know, and I know this is happening, that they're kind of looking at the leaders that have, you know, are in like phase 3 and phase 2 to kind of help define that. The things that are in late stages are mostly focused on FMT. So that's a little bit different, but it's still the same challenges. And I think they're also pushing back to say, you don't need to just even tell me what other bacteria in there. Now you have to check for things like viruses. So I think that the COVID pandemic, as well as some of the challenges the FMT folks have had in sick patients, are going to expand that you really need to have a very high level sensitivity to prove to me that there's nothing bad in there, that you're only putting in good stuff. So I think it remains to be seen. I think they don't have the right answer. And so the question is, you know, how much is good enough?
It's a big question, right?
Yep.
Nini, how has your experience been? I know you've gotten into clinical trials, so you must have been working with the FDA. Tell us about your experience.
Yeah, we are actually not working with FDA yet. Potentially, we might have to. Currently, the clinical trial, and after we consult with the FDA, we are mostly going towards to the state level of regulation. So now that's what we are trying to establishing with a CLIA or CLIA. So with the platform of MaMome, what we do here is not going towards to the drug therapeutic section at this moment. So we are more towards to a well-being device, or towards to the diagnostic area that essentially once we collect enough of data and make enough of a result to show them. But at this stage, we don't really have to touch with the FDA. But from what I heard, as Karen said, it's really how big and what kind of sensitivity of the data and what kind of protocol and method. Like there's no standard out there. And although FDA did mention about go check out 23andMe because they're sort of similar, but that is still not same as what we really doing with a microbiome. So that's just kind of feedback I got it back from a startup perspective view.
Interesting. Well, because you're not being invasive, so that you can.
Yeah, we can get away from that, I think, for now. Yeah,
that's great. Raj, Shetty, would you like to put on your camera and say hello? And let's see if you have any questions, comments.
Hello, this is Raj. Sorry, I'm a little bit under the weather, but thank you, Nini and Karen. I think the last slide, or the last 2 slides Karen shared about the challenges, and I think that's very important for the folks at Microbiome, working in the microbiome space, to understand what the risks and challenges are, improving, which microbes work and which do not. And, in my experience so far, I guess one of the things also I would like to ask Nini and Karen is around the intellectual property. How do you manage that? Considering the microbiomes are, I guess, from a patent perspective, are considered as natural materials. How, as a company, as you move towards commercialization, are you handling intellectual property?
Thanks, Raj.
Yeah, I can ask that. I mean, Shoreline Biome is really only focused. We have a patent issued on our method of lysis. And then we have a patent pending on our use of the long amplicon for identification of novel microbes. So we're focusing on really just, you know, the methods of our kits, and then being able to enable folks to use them. And so, but I know I talk to our customers all the time in terms of the content. I don't envision that Shoreline Biome would get in the space where we're trying to patent particular biomarkers. But, you know, we are sort of focused on enabling particular customers to utilize the technology to do that. But I certainly think that'll be an issue and a question for the folks out there that want to develop diagnostics, especially in therapeutics.
Yeah, for mine, we actually patented the provisional patent based on the method and the algorithms that we are more, I guess, bioinformatic computer tool focused. It is the pipeline that we're building, and particularly the microbiome groups that's focused towards the pregnancy and breastfeeding women. So these were really where we are looking for to find, hopefully, essentially there's some biomarkers showed up once we have enough of the data.
Beautiful.
Thank you.
Thank you, Raj. So we kind of touched on this, but I think that the idea of, since we're talking about moving research, you know, into actual practice, and it's not overstating that it's a complicated market and commercial platform. So can you each talk a little bit about challenges that you're seeing as you commercialize and how you're addressing some of those? I know, for example, Karen, you're working internationally. There's some unique features to working in that way. Go ahead.
Yeah, I think our biggest challenge has been limited marketing ability. I think messaging has been the hardest part for us. So we have great salespeople, but messaging is challenging, and trying to understand, you know, what customers' needs are in the science, as well as trying to message, you know, how you differentiate from what's out there. That's certainly been one of our key challenges. And then trying to do it in a pandemic when we're all at home. We do have international distributors, and also I think education has been difficult as well. What we've learned is that different regions are using the microbiome and the research very differently. And I think we've learned that. And so the messaging needs to be differently. For example, in Australia, majority of the microbiome work, not all of it, is very agricultural focused, whereas in APAC, it's more clinically focused. And so we've sort of had to learn that that's a very different customer segment, which has different sample types, all unique, and then different needs on the analysis. And then coming in where the majority of the customers have been using the Illumina short read technology and trying to educate them on the advantages of a long read technology, they also may not have access to, and having to rely on service labs is also, that's sort of a challenge, and we have to develop very close relationship with the platform providers. So that can be a challenge, is when you're dependent on a certain technology and platform, and trying to stay ahead of the puck on that.
Nini, what kind of challenges are you seeing?
I think because our sector is really focusing on the clinical application, and especially towards the OB/GYNs, I found the biggest challenges is there that you know, without the people who's in this community, not that many people actually really understand, physicians included, that what is microbiome and what reason research has been published. So it took a really, it took a lot of, I guess, time and then focus to really convince, and the OB/GYNs, here is a research that could actually be beneficial to the pregnancy and infant in the long run. That's definitely one of the challenges. And So from the research, I find out OB actually has the highest malpractice in the OD, is even higher than the anesthesiologist. So my clinical OB investigator, she pays $1.5 million for her malpractice just for the OB sector. And then, so she decided in this COVID happened, she decided to cancel her OB sector, so she doesn't have to pay for it. Then she can just, do the GYN, so her malpractice insurance could go down. So these are all essentially impact to the care of how to provide to the pregnant mothers in their trimester, and how they charge fees, and so essentially how they get being reimbursed. Because their malpractice is so high that they're obligated to see that many patients so they can run their clinic. And then with that amount of the time, how can we spare our time to really make them to talk to the patients that here's a clinical trial, you should really try it out. I found that's like really number one primary challenge. And also on top of that, come back to the end users, which is the pregnant mom, how they're really focusing on the nutrition takes. We want them to follow at least 4 weeks of our recommendation diet, but are they really going to follow? How are we going to find out if they have did it or not did it? And then especially in this special period of the life, that their family, everybody's involved. So this type of a monetization and collecting sample, I find is incredibly challenging. It definitely requires like the PI, like myself, to really run that and then understand from the physician, from the patient, and from the scientific aspect. So that's just brief to challenges. And then the third one I could think of is because the nature of pregnancy and infant, that how everybody's really focusing on care about them, that especially our test or the nutrition report has to be super regulated and understanding what are we providing. So these are the barriers, I think, and challenges for us to go to the market, even though finish the clinical trial. So we are really hoping that the result will come out great.
Well, thank you. That's terrific. So we're going to shift to other people's questions and comments. We have a really interesting question in the chat direction here. Have you considered, this for both of you, have you considered multiple applications? What do you want to do next to validate biomarkers? It may be possible to open more opportunities through creative international collaborations. Well, we know that's true, but I guess what are your next? Let's talk about biomarkers. What are you doing next to validate biomarkers? I think Karen, I heard you saying you'd be working with your clients on that.
Yeah, we have a couple of customers who have sort of expressed an interest in sort of moving it more towards a focused assay. We've done some work in the NICU, as well as with what we call, being able to determine if your NICU is clean and how the microbiome can you detect pathogens in a certain environment and track them within a care setting. So that's one application area that been looking at. We actually have a grant out with a collaborator. One of the things we're looking at next is, we've done the detection of the DNA in the wastewater, but the COVID in the wastewater is also another area. So we have a project looking at RNA and being able to detect not just microbes, but viral pathogens and combining that into more of a screening assay. So it's more of a screening focused. And then a couple of other customers who want to maybe focus on a subset using next generation sequencing, possibly on something like the Oxford Nanopore platform, and being able to do field-based screening as well.
And Nini.
We are still really early in the stage. So I wouldn't, we haven't really looked into it until we have a big first gathering data together. But we do have the intention, and we do particularly looking for the biomarkers for preeclampsia and gestational diabetes. These are the things that we've been tracing. from basically trimester 0 all the way to trimester 3. So hopefully there will be something promising to show up, but that's where are we right now? Thank you.
Well, I couldn't say it better myself. I want to thank everyone for coming. Everyone have a wonderful and safe holiday season. Thank you so much and take care.